Well, hello there, biology friends. Welcome to topic 3.3, meiosis. In this lecture, we're going to go over the overarching point of meiosis. But meiosis sounds awful lot like mitosis. And if you remember mitosis, it has all those stages. And so we will do those stages in class. I'm not going to have you in lecture. Actually write down the stages of meiosis. We'll do a chart in class so that we know. But if you remember if we want to go back and review mitosis, that is actually a good reminder because meiosis is just mitosis twice, right? We're looking at genetic variability. Oh, by the way, first period, who let me walk around with a hole in my elbow? I just went to go put my hair back in a ponytail. What is this? How come no one told me? I don't know about you guys, first period. All right. Meiosis 3.3. Let's do this. Okay. So we're going to be looking at why, the overarching question is, why are people so genetically diverse, even if they are coming from the same two parents? So if the, simple story is I get half my DNA from mom, half my DNA from dad, well, then how do brothers and sisters look completely different, right? So it must be a different half that I'm getting from mom and dad. And so siblings, cousins, all those aunts, uncles, why even within the same pool of genes are we getting so much diversity? So going to look at the mechanisms that cause that diversity. Again, the stages of mitosis, we'll look at how these sex cells or gametes are created, and then some things that happen later in life that eukaryotypes. Yeah, which eukaryotypes? Later in life that could be are more likely to cause I'm going to say chromosomal abnormalities and karyotyping. All right, let's get started. So the sexual life cycle, diploid and haploid, lots of information here. Let's kind of scan through and figure out what do we really need to write down here. Okay, so sexually reproducing organisms. Remember that sexually reproducing just means half and half come together, egg and sperm, versus asexual, exact copy, binary fission, those things. They inherit information from both parents, right? So we get half and half. So the resulting offspring ends up in diploid, right? So offspring equals diploid. So I get two halves coming together. Yay for math, right? So haploid equals, oh, what is that? There we go, diploid. Whoa, why? Let's go clear. Okay, I don't know why I did all those funny things, but apparently I can't do math on the screen. So pretty simple concept, right? So I've got two haploids, egg and sperm coming together. But in order to maintain that chromosome number, they must have all 46 chromosomes. So if all 46 chromosomes are not present, if there's any genetic abnormalities, we usually do not see the egg and sperm either develop or develop very far, right? Most miscarriages are because of some type of genetic abnormality where it starts to develop and the mechanisms within the cells say, nah, this is wrong, so it does not continue to develop. Some of them do, most of them don't. Okay, so when I get those two haploid cells coming together, which are called gametes, that creates a cell that has all 46 chromosomes. And from there, that one starts to multiply and differentiate into a new organism, right? So one single cell, egg, sperm comes together, starts to differentiate into a lot, exact copies. And then going back to what we talked about, stem cells, the ability to become anything, right? That happens almost immediately. Okay, so the offspring has a genetic combination of both parents, but is very, very different. So we'll be looking at that in the next couple slides. Again, scan through here, see what you have to write down. Don't need it all. All right, so here are my examples of gametes. I've already been saying this, egg and sperm, half and half. Egg and, sorry, I'm trying to clear this off too. There we go. And pen, clear screen. There we go. The egg and sperm contain half. Egg is, we're going to look at the end result, but eggs are very large. At the end of meiosis, I actually get 4 ending cells, but only one of them becomes what's called the ovum, right? At the end of meiosis for males, we're getting half and half, lots of different sperm with genetic differences. We have looked at, the flagella for movement, right? So all those different parts coming back right now. The process of meiosis differentiates the whole process is called gametogenesis. Gametogenesis, right? So genesis meaning to make, and then gametes is this process. These, do you need to know? No, we'll just keep that for the second screen. I'm sorry, the second one part. All right, so here's the process of meiosis. I am going to have you draw this into your margin, right? So draw this process into your margin. You can use your pretty different colors if you'd like, but what we are ultimately looking at is I have this cell here, right? So first of all, two divisions. Remember in mitosis, right? We looked at just that one division where I'm making an exact copy. In meiosis, I get two divisions that end up in half, right? And the end here is that we get four genetically diverse, right? So this first division here separates the homologous chromosomes, right? So first of all, I need to double my DNA. It's kind of fun. We actually times it by 2 in order to divide it by 2 because I need double before I go in half, right? So now I have my homologous chromosomes, and the first process gets one over here and one over there. So my homologous chromosomes separate, but think about your cells, you've got 46 of these in there. Right, so it's not just this one, but we keep it simple with the one. And then the second division here is what separates what we call the sister chromatids. Remember these chromatids here, connected here by the centromere, that protein, and so that we get half of the DNA in 4 different cells. So we double, divide, and divide that genetically, results in 4 genetically different cells. These all look the same right here, but we're going to talk about some of the processes that make them very different, right? So first division, diploid to haploid, and then the mitotic division of a haploid cell gets me here. Okay, our interphase, we're going to review just one more time, and then we're not going to do this part. So DNA. So remember the interphase, that the DNA, just to review, the DNA is replicated during the S phase, which stands for synthesis, and that the replicated chromosomes contain identical sister chromatids. Right, so the chromatids make identical copies. But the replication of the DNA means that it requires 2 divisions because I need half, right? So the second division separates them, created by interphase. So interphase, I get the doubling of it, and that first division, I get my sister chromatid. This is post-replication, right? So going back to what interphase is in meiosis, very similar to mitosis, the doubling of those. However, now we're going to be talking about chromatids, pre-replication and post-replication, and the things that they go through. All right, so we are going to skip, let's see right here. Should I even, hold on, I'm going to stop it. Okay, well, somehow I just stopped the recording. I've got holes in my shirt and I stopped the recording. That's not what I meant to do. I meant to pause it. Okay, so again, do not copy this down. Just kind of showing you guys what we'll be looking at in class, because we are going through those 2 divisions and you do need to know all the different stages, right? So let me scroll through them super quick. Don't write this down, right? We're going to be looking at prophase 1, metaphase 1, anaphase 1, telophase 1, prophase 2, metaphase 2, anaphase 2, telophase 2. Pretty fun, huh? So double the stages. Making sure that we know, we'll look through the PMAT again, so look through some mnemonic devices, and really going back to, like, if you remember the 1, what happens in metaphase, what happens in anaphase, what happens in telophase, just knowing that it happens once and twice. It's not as complicated as it looks, or as long as it looks, okay? This is what we talked about in the first part, but now let's go to how these different genetically distinctions occur. Okay. Oh, nope, not that one either. All right. Purpose of meiosis. All right. So here, let's start here. So if my purpose of meiosis is to make the ones that are genetically distinct, that doesn't happen, right? Because we need variation, right? So why do I need it? Variation, good for survival, right? We need traits, different traits, because environment changes. I need to make sure there's some kind of diversity so that some of the species survives. Remember, that is the goal of natural selection. Species survives, pass on to the offspring passes on. Okay, so we need to know these 3 different mechanisms that make them diverse, because you can't just copy and then divide and have things different. It doesn't work that way. So there's 3 different things we're going to talk about that cause this genetic variation. What are the 3 mechanisms which promote genetic variation? 1 is crossing over. Crossing over occurring between non-sister chromatids in prophase 1. We will look at that in the next slide. The second one is the process of what we call random assortment. You do need to know this happens in prophase and metaphase, right? And then that third one is the random fusion of gametes from different parents, right? Because we looked at the female creates 1 egg, but sperm males, we're talking millions, right? So all that genetic variation coming from male sperm cells, but just the 1 variation coming from the mom. Okay, so we're going to learn about these three in depth. And just remember that the goal, the reason is that I'm going to increase the chances that something survives if I increase variation, right? More biodiversity, more likely something is to survive. Okay, all right, so let's go on with starting with crossing over. I like crossing over. It's like I like when the vocab matches what the heck is happening. Okay, so I have my homologous pairs together, right? And what happens with those homologous pairs, remember that this is happening with all 40, all of the chromosomes. It's not just one. The homologous chromosomes connect via the chiasmata to form, right? Think of like the chiasm, right, where they cross to form what we call bivalents, right? Which are the tetrads. So these tetrads, tetra meaning 4, right? And when they come together, as that could be a chiasmata, right, to form those bivalents, we get them really close together. And what that allows them to do is their little legs here, you can imagine them crossing, whoop, and then they actually swap different, right, portions of their genes here, right? They actually swap different portions. These exchanges cause what we call recombinants or recombinations. And so I get two chromosomes that are completely different from my homologous pair. Then these two separate, and then I get 4 chromatids that are completely different, right? So these crossing overs happens at the same place in my chromosomes, or at the same location. So it's not like one's having more than the other. It's just swapping my different alleles or the different versions that they have. And this process is one of the ways in which I get some genetic diversity. Okay, so again, that is my one. Moving on to that, crossing over one. Again, we'll look at some things in class. The second source of variation is called random assortment, and that is in metaphase, right? Remember we talked about they meet in the middle. They all line up in what's called a random orientation. So there's an equal chance of the resulting gamete getting either of the pair. So it's not like every single time they meet in the middle in metaphase, they line up in the same order. It's random how they orient themselves. And so sometimes we get this chromatid, sometimes we get that chromatid, right? So because there's an equal chance of either one of those pairing up, humans have 23 pairs of chromosomes. So there's a 2 to the 23rd gamete combinations of all the ones that are already mixed up because of crossing over. Right? So the source of variation random assortment is that it's different. It's, the same half doesn't always go to the same side. So looking back at this one here, right, these ones all crossed over recombinant. They might end up in different gametes, and then because they're all switched up, so we've got variation among variation, right? So that third process is called random assortment. Okay. The third process is called the gamete fusion, right? I already talked about this briefly, but when I get the fission of those two, fusion of those two haploid gametes that result in the formation of my zygote, again, a lot of different variation in the sperm, the one variation in the egg that's made, I get the zygote that is randomly fertilized, and then I get a genetically distinct embryo. Zygote, embryo, fetus. Those are the stages, Zeph. Okay. And so when I see, do I have twins in here? I do not. Okay. When I have identical twins, what happens is when this process happens, when the zygote is formed, the cell immediately creates an exact copy, right? So I got a genetically identical twin. If I get fraternal twins, that means that there's 2 eggs being released. So I've got 2 eggs with completely different genetics being fertilized by 2 different sperms. So fraternal twins have the same genetic, distinctness as siblings do, right? So they're just basically siblings that are born at the same time. Identical twins, though, this zygote completely makes an identical copy before it starts to develop into an embryo. Okay, so that is the end of part 1. In part 2, we're going to look at the things that can go wrong with chromosomes, and then look at some karyotypes and how we're able to tell when things go wrong. Let's hope that I can put these videos together, and that tomorrow I can find a shirt without holes in the elbows. I will see you guys later.